material.
Thank you. So I'm talking about, when I wrote this abstract, I had it as larval Coregonines. That's before we finished all the genotyping. I'm going to change it to Cisco. We finished our genotyping. The vast majority of the fish that we genotyped are Cisco. It's only a dozen or so, or a couple dozen lake whitefish. So the stuff that I'm about to talk about, you need to have good sample sizes for to make it work, because these things are really good fun. Before I get into it, this work is a team effort all around. I'm a geneticist, but I work a lot with USGS in Oswego and at Tunison. So Brian Weidel's group here at T-Lobs is instrumental in getting out there in this tough weather, collecting all these fish. So I really appreciate all of their work. Amanda Actis and Ann Ropp at the Great Lakes Science Center in Ann Arbor are geneticists as well, and the way I genotyped these fish was with an assay that she developed. So she really helped get that assay up and running in the lab, and then of course all the people in my lab that did all the bench work, starting with my lab manager Vinny and technician Grace and the students that are here now. So thanks Joe and Colin. So our group is interested in addressing knowledge gaps in an adaptive management framework. So this is Paul Bonnell's paper that he published where he talks about the Coregonine restoration framework and doing research in this wheel type of approach. And we seek to prioritize research that aligns with the Coregonine restoration framework. So that means we are looking to understand population dynamics, so connecting habitat change to fitness metrics, and also conducting experiments. So here, like planning, it's a gap analysis, trying to understand what habitats really can affect fitness. Alex's talk, he had a great survivorship curve up there. We're talking about things that are super fecund, they make lots of babies, many of them die, and if we can have just a small effect on those early life stages, that could really mean something for the adults in the population. So that's why it's so important. And it's hard to study this stuff, right? We don't know. Our understanding changes through time. We are studying. What's the classic quote? It's studying fish is like studying trees, except they're invisible and they move, right? And so it's hard to understand what's happening in these systems. And so to understand mechanisms, you have to conduct experiments. And we have done that, or we have started to do that in this adaptive management framework. So we've done a habitat remediation experiment to evaluate how depth in interstitial spaces affects Cisco egg survival and spawning adult habitat use. I started working with Cisco in 2018 when I moved to Oswego, when Brian asked me to help genotype fish for Taylor Brown's work, where we looked at the spatial extent of Coregonines in Lake Ontario. And of course, when you look at that for Cisco, so we've got our colors here representing number of larvae per 1,000 meters cubed. You can see there's a ton of larvae that show up in Chameau Bay. And so that's why Chameau is the focus of conservation and restoration efforts and the focus of my talk. This is a slide that Brian Weidel put together, and I think he does a really great job at talking about visualizing what we're doing. So we took a habitat-specific life stage approach. We've got larvae, juvenile, adults here, and what we're studying is the other side of this circle. We're studying processes, so we're studying incubation, egg settlement, spawning, and at the stage of observation that we're looking at are larval emergence, and in this study, egg deposition. And I really love Alan's talk. That was like perfect for this. It helps me understand it. Again, I'm a geneticist. I'm a relatively naive person in the field, so it helps me understand the types of fish I'm getting. Sard does the genetics, while you guys do the actual fisheries work, getting the tissues to my lab. So this is a metal ring trap that we use to catch eggs from spawning Cisco in Chameau Bay, and an emergence trap that was developed by Brian's group. Very similar to what Alan was showing, right? And so she's already done a good job of describing those. So normal type time. So I'll just say we use those two things to study egg deposition and larval emergence. One of the things, so is speaking to egg deposition, when you look on the x-axis of this figure here, we've got depth on the x, and we have egg deposition on the y. And you can then have these different colors of different substrates that these eggs were deposited on. And you can see there's this signal here, about 2 to 4 meters. And in some cases, you have really big numbers in terms of the number of eggs deposited. And you can see a fair amount of muscle hash on there, a little bit of cobble and boulder. And so the question is, you know, is it substrate, is it depth, or both? And substrate and depth are not independent of each other, as depicted with this figure. Depending on where you are in Chameau Bay, you see different types of substrates. And so how do you understand what these fish are spawning on? Well, you conduct an experiment. You test this idea. So this is the ecosystem experiment BW and others have put together where we tested if adding interstitial spaces or substrates improves egg survival. And this is a photo from Joe Johnson depicting with these rock piles in Middle Shoal. So, you know, we're talking about Chameau Bay. This is Chameau Bay. These red dots here are all the, these are the larval emergence trap sites, and you can see them here. So we have these substrate addition sites at 2 meters, 3 meters, 4 meters, and 6 meters, and we have control sites right next door. And so at these sites, from November to December, we drop egg traps, about 50 per site. We also, in the springtime, mid-March to May, in this really nice weather, you know, getting out on the lake, look for larval, we set larval traps. And so in total, the number, sample size that I'm working with here are just under 2,000 fish, 100 traps, or a little more than 100 traps, and about 60 toes. So take-home messages from this work without the genetics, okay? Without the genetic pedigree stuff that I'm going to talk about. There was genetics involved in terms of the species. So we have this increasing evidence that coregonines are spawning is habitat specific. And we found that through sampling egg deposition and larval emergence, and we did that, we needed this appropriate gear to be able to get at that question, right? And then we also did this experiment, this ecosystem experiment, this habitat remediation, right? Like teeny species. What's the major driver of becoming a teeny species? It's habitat. So when you put good habitat in, what's going to happen? What we see is that both depth here on the X and substrate, the colors, matter. And so this is, if you have egg deposition and you have a metric of larval emergence, you can calculate survival. And what you see is that there's very little survival of eggs on the control sites, and much more survival, especially at the 2 to 3 meter spots relative to the controls. So I told you I'm a geneticist, so I'm going to connect larvae to families. And it's larvae really because you have enough tissue to actually get a good enough DNA extract to make this all work. So we're looking beyond larvae. When you look beyond larvae and you look at their genetics, they are telling a story about their parents. And so you can infer parents from the DNA of the kids, and you can infer family relationships. And this helps us improve our understanding of reproductive dynamics. It's an estimate. It provides an estimate. So we know that fitness is a two-part metric. It's survival and reproduction. We just talked about egg survival. And I'm going to layer in the number of parents. I'm not going to get into the fitness stuff, but we have that as well. We're going to estimate the number of parents that produce this set of offspring. And you know, BW stuff is great, but Cisco are super fecund, right? So what is BW catching when he catches eggs or larvae in his samples? Is it a lot of eggs from a couple females or a couple mate pairs, or is it a lot of eggs from a lot of different mate pairs? And that's what this is about. And one kind of highlight here, without getting into the genetics too much, we're talking about pedigree reconstruction without parents, which is a really challenging thing. You need a lot of information to do that, a lot of alleles, and the program that I use is it's pretty much the only game in town for pedigree reconstruction without parents. and it's called colony. The other thing I'll add is when you start understanding family relationships, like you get in this world with dyads or pairs of individuals, and you can look at where one full sibling is compared to another full sibling, or where one half sibling is compared to another half sibling. So you can start looking at connectivity on a fine scale of family groups in space and time. All right, so I told you we GT-seq this sample. This is like one of the common ways to do it. I mean, the point is you get hundreds of markers, you get really good likelihood scores for the gene types you're confident in them, and it's a standardized set of markers across all individuals. So a good quality data set. And Amanda, thankfully, developed one for species ID in Coregonines a few years ago. And so what we did is use that in the context of pedigree reconstruction. So her lab kind of got us off the ground, thank you, and my lab finished it up. So we reconstructed the pedigrees. We estimated the number of parents at specific spawning sites using the larval emergence tracks. But now Ellen's got me really thinking about what the heck am I looking at? So I gotta think about what you said here about 40 minutes ago. And I'm never good at thinking that on my feet. So we'll see what happens here. So toe samples were used to estimate the number of parents for the entire bay, and you actual fisheries biologists need to tell me how crazy my estimate is when we get done with this thing. So I'm saying number of parents, right? What is number of parents? I want to say it's not census size, right? Because let's think about lake trout. Like, not every adult in the lake trout population is spawning every single year, right? It's not effective number of breeders or effective population size. Those are conservation genetic metrics, and they're affected by sex ratio, they're affected by variance in reproductive success, and they're grounded in a rich theory. This is different. This is in between those two things. This is the number of parents, and it's a rehash of community ecology literature. So we know that as you increase your sample size and as you're trying to survey a community, you should expect to see more species, right? And you can form a species accumulation curve. And then at some point you should expect an asymptote, that the more you sample a system, the more species you detect until you're done, until you've actually detected them all. And of course we're never there because they're super hard to sample, and people have developed non-parametric methods to estimate the number of species in a system. This one I'm talking about here is the Chow estimate. So it looks at the number of species detected just once or twice, and the relative frequency of those. And it inflates the number of, the math inflates the number of species that you actually have in hand by that ratio. And it counts for sample size too. But we thought, well, that's what's happening with larvae, with their parents. So as you sample more larvae in a system, you're going to pick up more and more of their parents. But at some point, if you sample the system enough, you should have a saturation of the number of parents inferred. So we showed in a molecular methods in ecology and evolution paper a few years ago that this method can work in this type of system. All right, so let's get into the results. I also mentioned toes. Sorry, I've said toes a couple times. I just want to say in Middle Shoal, they did ichthyoplankton toes over the shoal and throughout the bay. And so that gives you a better representation of all the offspring that are in the bay as compared to just in Middle Shoal. And that's why I use them to estimate the number of parents. I'm going to skip this because I'm running out of time and I'm going to get right into the results here. So substrate added on the left, control sites on the right, site 1, site 2, site 3, site 4 for both the substrate added and control sites. And this is our number of parents, our estimate. And what you see is, well, one thing, wherever we looked, we saw hundreds of adults. We inferred hundreds of adults produce the larvae that we caught. Okay? The other thing is, you can see it's redder over here than over there, right? In that there are more parents, parents estimated to have spawned on the substrate additions. And so this number of parents estimated per site corroborates the age survival estimates. And then we're going to dig into this a little bit deeper. So this is a pairwise figure, okay? So we've got control sites here, 1 through 4, substrate sites, 1 through 4, on the X and on the Y. and I have full siblings and half siblings in the panels. And what I'm showing you here is counts of full siblings, and on the diagonal is within site comparisons, and off the diagonal is between site comparisons. And here, I know this is a lot, and so feel free to stop and ask questions here. But here, this is all within control sites and among control sites. Within substrate sites, among substrate sites, and then between substrate and control sites. And where do you see full siblings? You see them within substrate sites as you would expect them to be. Not, this is full siblings, right? So the odds of those parents mating in another substrate together are relatively low. So you should expect a full sibling signature stronger here. You also see half siblings, so similar type of picture here for the half siblings. This is it, Ellen, right? I'd love to hear what you have to say about this, right? This is substrate site 2 right here, and this is substrate 1, right? And so there's this interplay between half siblings. So one parent mated there, and then what I interpreted as they left and went and mated with another site. But how much does a larvae move? How do you interpret that, right? But the other thing about the movement that I was thinking about during Alice's talk is, what's the distance from here to here? So I got to go calculate all these pairwise distances and kind of think about this a little bit more in terms of the larvae's behavior. And I know I'm pretty much out of time, so I'll just say we look at the toes, right? So toe samples describe larvae in the main space and time, and we use to estimate the number of parents. I already talked to you all about all that. Now, I can give you an estimate. I can calculate the numbers. The question is the number good? And I don't know, right? This is a new method, and it needs to be vetted and tested, and so you should be wary of new methods, right? And they need to be ground tested. So one thing to consider here is 100 random samples. So what we had is 1,000 fish. And I took 100 random samples of those 1,000 fish, sampled from a uniform distribution from 2 to the top, and with no replacement, and I ran the colony runs again, okay? So when I ran the colony runs again, I then estimated the number of parents using the child method, and then I plotted them, right? And so if we did a good job of sampling larvae in the bay to detect all the parents, you should expect to see an asymptote, right? You should expect to see a slowing of the slope through time. All right, here we go. And so what did I actually find? Larval sample size on the X. So there's different colors here, and this is estimates of parents on the Y. My total estimate is a little over 1,000. Is that crazy or not? I have no idea. I'll have to ask you folks who know more about the system. But what you can see is that, one, I'll focus on the light blue dots here. These are the resampling without replacement among all the toes. And you can see as we increase sample size, so too does our number of parents estimate. The dark blues are actually within toes, like how much bang for your buck can you get for a single toe in terms of estimating the number of parents. And you can see it's not very much. It's all over the place, it's pretty low, it's certainly biased low. So I have the take-home message here is at least 1,000 ciscos successfully reproduced in Chameau Bay in 2023. And now it's your turn to tell me I'm crazy and wrong. What's next? So, like, when you get into this world of sibship, it turns into dyads, and turns into pairs of individuals. If you got 1,000 fish, every fish gets compared to 999, and so the data sets just blow up like really quick. And then they get into like weird independence issues and whatever. But one thing that I'm interested in is BW's looking at flow in the system, right? So thinking about larval drift. All right, here's Middle Shoal, right? And this is work with a crew out of UB. And so we have sampled the crap out of Middle Shoal with all these larval traps, right? So that's our centroid. We've also sampled all throughout the bay using ichthyoplankton toes. And so you have like how connecting full siblings and half siblings from Middle Shoal on our traps to the ichthyoplankton toes and trying to describe what environmental variables explain, or test, I should say, test what environmental variables explain where they are in space and time is something we're after next. I'm going to repeat this for the 2024 larvae, and then I was thinking about this and getting all excited about it, and I was texting BW and I was saying, hey, we should start getting adults from your prey fish survey, right? He's like, oh, I've already done that for like 8 years. So I'm super excited to gene the adults, assign them to the larval cohorts in Chameau because it's one of the major games in town, and use it in a CKMR framework to actually estimate census size. So I'll leave it there. I know I'm over, so thanks for listening to me blab.